Hello friends, welcome back to my channel, and today in this video we are going to study about the functions of an antibody. In the last video we learned about its structure, and today we are going to deal with its function. So as you remember from the last video that this is the basic structure of an antibody that consists of a heavy chain, this dark colored, and this light colored is L chain. Now in this light chain we have two segments, one is variable and one is constant. Similarly, even in heavy chain we have variable and the constant. In some antibodies we will have three constant regions, and some we have the four constant regions. And all these light chains and heavy chains are connected with disulfide bonds. And center here we have hinge region. So now when we add enzyme pepsin, so what it does is the cleavage site of the pepsin is below this disulfide bond of the hinge region. So what happens? It will remain with Fab. In the last class only we learned Fab means the fragment of antigen binding. That means the variable part. For whether it is a heavy chain or a light chain, the fragments that bind with the antigen is called the variable fragment, VL or VH, right? So this is one set of VLVH and this is the second set of VLVH. Hence it is written Fab AB square. So because the cleavage site of pepsin is just below this hinge region, so we get Fab region, two Fab regions and FC fragments. FC means fragment of crystallization. We learned in the last video itself. When we digest the same antibody with papain, so the cleavage site of papain is here, just above the hinge region. So now you tell me, if I cleave here, the fragments obtained will be like this one fragment, like this another fragment, and like this the third fragment, because the cleavage site is here. Correct? So we will get Fab region separately, two Fabs. With pepsin digestion, the two Fabs are joined. Here we will get separate Fabs and this FC region. What will happen if we digest it with mercaptoethanol? Mercaptoethanol is a reducing agent. So it will reduce. Now disulfide bond is present between two cysteine molecules. In the reducing environment, the bond will break because now the S will get reduced and form SH. The bond will break. So once the bond will break, because the light chains and heavy chains, and from this hinge region, both the heavy chains are connected with each other. So once all the disulfide bond is broken, we will get one light chain, one heavy chain, second heavy chain, second light chain separately. So we will get like this. Clear? So depending upon the enzyme used for its digestion, what is the property of the enzyme or what is the cleavage site of the enzyme, it will give different products. Now antibody, it fights with antigen, but my antibody, that is the antibody present in my body, work as an antigen for antibody present in your body. So this is me and this is you. Antibody present in my body will fight with the antigens. But because this is the antibody of my body, the same antibody will work as an antigen for your body. Okay. Now FC and FAB we already learned. So this is called antigenic determinants. That means the markers or the structures or the parameters of the antibody that function as an antigen in any other person's body is called as antigenic determinants. So antigenic determinants are the biomarkers, or you can say these are the markers present in any foreign substance. So antigen means what? Antigen means anything foreign to our body is antigen. So our body will react against it. So how the body will react, how it will bind with the antigen, how it will recognize it as an antigen with the help of antigenic determinants. Similarly, my body antibody is foreign for your body. So for your body antibody, this my antibody is antigen. So it will recognize it with the antigenic determinants. Means for my body it is not antigenic determinants, but for your body it is an antigenic determinants because this is the antibody present in my body. So the antibody which is present in my body is foreign to your body. So for your body it is again an antigen. Now this is the table which shows the type of antibody and what it recognizes. Now first is anti-Fab. So with the name suggests, anti-Fab. That means with which it will bind? Fab. What is Fab? The variable part. So it will bind with the variable segment, the Fab segment. Anti-FC. It will bind with the FC. Now next three is important: anti-isotype, anti-allotype and anti-idiotype. Anti-isotype means these antibodies IgA, IgM, IgD, IgE and IgG. So these are isotypes of each other. IgA is one isotype, IgM is one isotype, IgD is one isotype, IgE is one isotype. So these are all isotypes. What is allotype? Allo, that means allelic. Allelic specific. Allelic means IgA of my body will be different from IgA of your body. So this is allelic specific. That means difference within the members of same species. So the my body IgA will be different from your body IgA. So that is called allotype. What is idiotype? Idiotype means my body IgA, which is specific for antigen 1, will be different from my body IgA, which is specific for antigen 2. Clear the points? Isotype means the different classes of antibodies. Allotype means difference in same antibodies within the members of same species. Idiotype means I, idio. Remember, individual. It is the individual's antibody which is different from individual's antibody. So IgA, which is specific for one antigen of my body, will be different from IgA of my body, which is specific for antigen 2. So that is an idiotype. So these markers are present in the variable regions because it is binding with the antigen. It is specific to antigens. These are antigen specific antibodies. So the markers for this are found in variable region. Isotype marker and allotype markers are found on the constant region, but idiotype markers are found on the variable region because these are antigen specific antibodies. Now let's understand some specific function of light and the heavy chain of antibody. So if we talk about this Cl and CH1, that means this part. Now as I gave you the example in the last video, that this hinge region consider as your elbow. Now if I throw a ball towards you, depending upon the size of the ball, either your hands will be far apart or you will bring both your hands close to each other. Similarly, antibodies also, with the help of hinge region, to hold the antigen, they will move their hand to hold the antigen. Now CH1 and Cl1, you can say them as forearm, and this is the palm. Variable region is the palm. Now you will hold the ball with the palm. Now what will happen if you don't have this forearm? Directly your elbow part, and with this elbow part you have your palm attached. Do you think you can able to hold the ball? No, right? So the arm is very important. The forearm is very important. Correct? It facilitates the extension and maximizes the ability to bind or to hold a ball or to bind with the antigen. So the forearm is important. Right after your elbow, if you have palm, you cannot hold anything. So you need to have forearm because that will give you the extension and the possibility to move your hand so that you can hold the ball. Similarly for antibody, this forearm, that is the Cl and CH1, works. Coming to the hinge region, already the function I explained you. Hinge region is rich in cysteines because it helps to bound two heavy chain with each other. It is also rich in proline residues that helps to provide rigidity with flexibility. It should not be that flexible that it breaks. So it should have some rigidity also along with the flexibility, because of which it is also rich in the proline residues. Coming to the carbohydrate chain, if you remember from the structure of the antibody video that we have carbohydrate attached with the antibody, that helps to make it aqueous environment accessible and also helps antibodies to bind to complement components. Now coming to the carboxyl terminal end, that is the Fc region. So the antibodies are of two types. We learned already that B cell secretes antibodies. Now B cell, the effector B cell, will secrete antibody, whereas the memory B cell, it will bind the antibody to its surface and remain bounded form. So antibody has two forms. One is in the secretion form, another one is the bound form. The secretion form means it is going in the blood or the body fluids. So it should be aqueous soluble. So the amino acid should be polar in nature, hydrophilic amino acids. Coming to this antibody which is bound with the B cell. So it has three segments, as you can see. Suppose this is your cell membrane. This is the antibody. So now as you can see, it has three segments. This is one part, the external part. It should be hydrophilic because outside environment, outside of the cell is the aqueous environment. Then you have this part which is embedded in the membrane. So that should be again hydrophobic. And this segment which is in the cytosol. So the membrane bound antibody should have three parts. The extracellular should be hydrophilic, the transmembrane should be hydrophobic, and the intracellular, that is, it should have a cytoplasmic tail, which should also be in a soluble form because the environment inside the cell is aqueous, environment outside the cell is also aqueous. So this is about the function of each and every parts of antibody. The function of variable part, the function of hinge region, and the function of constant region. Now let's see the structure of different antibodies, this different isotypic antibodies. So we have IgM, IgA, IgD, IgE, and IgG. IgM, it is found in blood and lymph. So it constitutes the first line of defense. Any infection that enters our body via blood or lymph, IgM will capture it. IgA, it is called secretory antibody. It is present in all our secretion, that is saliva, tears, mucus, breast milk. So it provides protection from the infection that enters either through ingestion or through inhalation. The antigen that enters our body through ingestion or through inhalation, the IgA will take care of it. Next come to IgD. It helps in B cell activation and maturation. Next is IgE. IgE, it mainly functions in allergic response. So when it is responsible for allergies, that means IgE is found in our skin, lungs, mucus, because it is responsible for allergic infection. IgG, it is also present in blood and tissue fluids. So because it is present in blood and tissue fluids, so IgG can cross the placenta and can enter the fetus body from mother body. So when the fetus is inside the mother womb, the antibody that provides protection to the fetus is IgG because it is present in tissue fluids and it can cross the placenta. Once the baby is born, the mother's milk provides protection to the newborn baby because breast milk contains antibody. And which antibody it contains? That is IgA, which is present in all our secretion. After that, it is taking care of IgM, which is the innate immune antibody, we can say it, which is present in our blood and lymph. So IgD as such no function in protection, it helps in B cell activation, and IgE, it function in the case of allergic reactions. So that's all for the video about functions of antibodies. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.